Hello and welcome to this Chemistry World webinar. Analytical impurity standards, minimising project risk and avoiding common pitfalls. I'm Philip Broadwith, Chemistry World's business editor. Just before we start the webinar proper, I'm going to have a couple of minutes just to go through some of the features of the GoToWebinar platform that we're using. It's an interactive platform, and that's really important because what we'd really like is to get the questions from you, the audience, to our expert speakers. At any point in the webinar, you can ask your questions and we will put them into the Q&A session at the end. There is a box near the bottom of your GoToWebinar panel. By default, that's on the right-hand side of your screen if you're using a PC, but it might be somewhere else if you've joined from a Mac or a mobile device. Just have a look around, find that questions box, and that's where you should ask your questions at any point. They are an absolutely crucial part of the webinar, so please don't wait until the end to put your question in. It really helps us with question scheduling if you can get them in nice and early. If you do miss any part of the presentation, or you just want to watch it again on your own time, pick up the details and links and resources that we've included, then you will get a link to the recording that you can come back to by email in a couple of days' time. And to say thank you to everyone who's attended the webinar live, you'll also get a certificate of attendance in that same email. So today's webinar is about analytical impurity reference standards, which are a crucial part of assessing the quality of processes across a whole range of sectors, from pharmaceuticals, forensic science, and environmental analysis. We'll look at some of the challenges of producing high quality reference standards, some of the potential problems you might face while buying and using them, and we're going to include a case study of the consequences of using low quality reference materials. We have worked with Toronto Research Chemicals, which is part of the LGC group, to bring you some information on this that we think you'll be interested in from absolute experts in the field. Later on, we'll be hearing from Tony Rantanen, who is a quality manager at TRC based in Toronto. He has a background in synthetic chemistry and extensive technical expertise from working at TRC for over 8 years. But first up, we've got Joe Lackey, who is a technical specialist at LGC based in the UK, and he's going to be taking us through some of TRC's approach to creating high quality reference materials. Over to you, Joe.
Thanks very much, Philip. Can you hear me okay?
Yeah, we can hear you, and we've got your slides up there on the screen now. So I'm going to let you get on with it.
Brilliant. Okay. Well, good morning and good afternoon, and welcome to this webinar on impurity standards, avoiding the pitfalls. So in this webinar, I'm going to give a brief introduction to Toronto Research Chemicals, and then moving on to some thoughts from a customer perspective about the importance of quality and what this means in terms of manufacture and people's research. So, next slide, please. Yeah, and then I'm going to go on and discuss the results of an independent study carried out by the Broad Institute in the US into the quality of drug testing standards on the market, which really does give some cause for concern. And then moving on, I've been working with Tony and team, and we're going to discuss TRC approach to quality and how we've designed our systems to ensure that we deliver a product which has the right structure and purity. And of course, with quality, structure ID is really everything. So wrong structure means wrong ID. And so Tony's going to talk about the challenges in terms of quality of supporting a 150,000 molecule catalog, and then going on to talk about some of the more unusual ID techniques and how to onboard these and ensure quality. And then finally moving on to some case studies, looking at some of the challenges in the shortfalls in the literature and how this can impact on consumer choices for standards and indeed manufacturing the right impurities as well. And then finally, we have a case study from our customs services department, which really illustrates some of the problems that you can run into when using standards which haven't been appropriately vetted. Okay, and then of course, we're happy to address any questions. Next slide, please, thanks. Okay, so to give a little background into the company, TRC was founded by the Dime brothers, David, Larry, and Charles in 1982 as a spin-out from Toronto University when David Dime observed a lack of research chemicals in the market. And Toronto Research Chemicals have now been providing complex organics to the research community, pharma, and also applied markets for now more than 40 years. And we synthesize more than 100,000 unique chemicals. So in 2022 now, we've got more than 250 PhD and master's level chemists working across 17 production sites. So David's slogan sums up the company nicely. If it can be made, we can make it. And we're currently in the process of developing, expanding our site, from 120,000 square foot, and there's a new site being built in Toronto to keep up with demands. So next slide, please. Thanks. So since this webinar is on a theme of quality, I thought to start by discussing the types of drug standards on the market. So what's available from LGC in terms of TRC, and also our sister company Micromol, and then leading on to the main uses for these types of standards. So of course, the major factor which should determine your choice of internal reference standard is the end use you have in mind. And there are two main types of analytical use, so qualitative and quantitative. So TRC provide qualitative products as standard, and I'll talk a bit about TRC robust approach to identity and purity testing later on. So the main difference for a qualitative versus a quantitative standard is that qualitative standards are provided at accreditation ISO 9001, as opposed to our quantitative standards from Micromol, which are provided from a lab that's certified to ISO 17025 standards. And these can be used, these type of standards can be used to demonstrate compliance to relevant regulatory frameworks such as ICH, as well as GMP and GLP. And from an analytical point of view, these quantitative standards can be used for exact quantitation and validation of analytical methods. So with Micromol, for example, residual water, volatiles, and inorganic contents are determined as standard, as is the norm for a quantitative standard. Whereas this kind of testing is not always part of the standard menu of testing for TRC qualitative standards, but of course, we can provide any test on a custom COA to tailor the standards to other things that you might have in mind. So to go through some of the end uses, so for TRC, these are really very varied, and we have polled on this question as well. But the main ones are system suitability tests. So often for validation of in-house analytical procedures. So this is when you have, say, an API and impurity, and they're eluting closely together. So you would want to sort of recreate this and mix, API and impurity standards and run these before your real samples just to validate and test the system. And quite a popular one is peak identification. So this is for identification of unknown peaks. So often you have a tentative molecular structure, and you want to compare this with an analytical standard that's been qualified by a couple of different orthogonal methods. So more than just mass spec, often proton NMR as well. So additionally, things like validation of specificity parameters. So just to validate your API assay, often by spiking in an impurity standard to, just to confirm it doesn't interfere with the assay. So TRC have a large range of APIs, impurities, and drug metabolites, and also things like stable labeled isotopes. So as discussed, we also provide the quantitative reference materials from Micromol, and these are APIs, impurities, and excipients as well. And common uses for these types of standards would be quantitative applications, such as obviously direct quantitation of an impurity using a reference material, and also things like kind of determining relative response factors in order that you could then quantitate your impurity from this. But these types of quantitative approaches are sort of beyond the scope of this particular discussion. but we're happy to, you know, kind of if you put things in the chat, we're happy to address these in the questions. So next slide, please. Thanks. So why is quality control so important in chemical synthesis of drug testing standards? Well, speaking with customers, it's clear that either, you know, speaking from a manufacturer or research end use perspective, quality control really permeates our life in every respect. So if we're buying a chemical, the expectations are that what's on the label will also be in the bottle. So if the identity of the chemical is correct and the specifications and purity are also correct is the important thing. And this impacts on the way customers perform research and manufacture product. And without quality control, of course, the identity of the molecule may be incorrect, the purity may not meet specifications, and of course this will impact on everything that I would do as a customer. So to ensure quality control in chemical synthesis, we have to ensure that we get the right structure. So wrong structure means wrong ID. And if a customer has been using this as an analytical standard for manufacture or research, then a wrong or poor quality standard could completely invalidate the work that's being carried out, leading to wrong conclusions, a loss of time and money, and of course, you know, most importantly, potential safety concerns as well. And a recent investigation carried out by the Broad Institute really underscores some of these issues. So the Broad Institute, they purchase you know, around about 8,500 API analytical standards, and they reviewed the quality of these. They analyzed them independently and found that in fact 29% of these standards were below the stated purity, and often significantly so, and they were, you know, way lower than 85%. So it doesn't seem a big leap to assume that impurities, you know, there may be a similar issue with these as well. Okay, thanks. Next slide, please. So from a TRC perspective, what systems have we put in place to ensure product quality? Well, TRC have developed a robust quality management system designed to ensure that you always get the correct molecule and with no doubts about the quality. So these are set in place to ensure that you get the right structure at the stated purity. And I've had some interesting conversation with Tony's team and others at Toronto Research Chemicals, you know, about the challenges faced in supporting the breadth and diversity of the TRC catalogue. So we have more than 150,000 products, more than a third of which are routinely in stock. So we've got compounds where the structure appears very simple, very trivial, and we also have compounds that are unstable to light, air, and other things as well. And these range from small organics to complex organics, polymers, and stable label isotopes as well. And this, of course, poses a daily challenge to the team on how to pivot from analyzing these relatively simple and stable compounds to modifying work accordingly to accommodate these more difficult challenges. And in terms of, you know, when we hire new analytical chemists, we always try to ask quite a lot of organic chemistry related questions. So we require analytical chemists with a very strong organic chemistry knowledge. As an example, to underscore some of the specific difficulties, so we may have a pharmaceutical impurity compound, but we don't have existing internal processes detailing how to test this compound in the same way as a pharma company. So whenever we get one of these cases, our chemists have to think, well, based on the structure, how do we handle these compounds? You know, and based on the structure, do we need to handle it? Is it moisture sensitive or light sensitive? Do we need to handle it in an inert background, in argon or nitrogen, for example? Based on the structure, we have to think, what tests do we need to confirm to confirm the structure? Do we need to do an ID test, and which type of approach should we look at, you know, to resolve this? And then we think, how do we need to determine the purity? If we use HPLC, or if we have to do a titration, so whether we can release it or not. So this is a big challenge in terms of quality control analysis at TRC. And then we have a quality assurance group as well, and they have to collate the data and determine, if we've confirmed things successfully or not. And, often this can be challenging, especially in the absence of, sometimes these are new compounds and, there's no existing literature. Okay, next slide, please. So how do we set up the team at TRC in a robust way to ensure we always deliver a quality product? Well, we've got several teams working together to ensure compound release. So the chemistry planning team, they plan and study and investigate synthetic routes to manufacture of a molecule. So they combine details from the literature with their own experience and thoughts and knowledge, and then design a plan of action. And we have our production team who manufacture the molecule, and then after that we turn to our synthetic chemist team, and they kind of perform various analytical impurity tests, and then finally after this we go to our quality assurance team, and this team goes through all the data, comparing the compound data with the literature and according to their own experience, to ensure if the identity impurity is truly what it's supposed to be. So we've got multiple teams responsible and working together to release the correct compound at the desired purity, and we feel that we've really put a robust system together in place to ensure quality. So in terms of the timeline, well, this really depends on the availability of raw materials, and also the complexity of the synthesis. And of course, anyone who's done a lot of synthetic work will know that even if there's a lot of literature precedent, when you're trying to repeat a synthetic procedure in-house, it doesn't always turn out how it's supposed to. And this, of course, poses challenges to the synthetic team, and sometimes timelines have to be extended. Now and then some reactions simply don't work, and of course we've got our talented chemists who will do troubleshooting, problem solving, and they'll endeavour to come up with an alternative route to manufacture. Brilliant. So next slide, please. So as you can imagine, the internal expertise and equipment resource to support the type of quality control associated with this 150,000 molecule catalogue is significant. And for the identity tests, it's important to confirm the identity of the analytical standard, not just with one technique, but with a sensible combination of methods. So in most cases, this means proton NMR and mass spec, and perhaps infrared spectroscopy. So we also have a UV. We test the specific rotation as necessary for chiral compounds. And if the structure is too complex, or if it's brand new, sometimes we need to collaborate. So we might do an X-ray in collaboration with Ottawa or Toronto University. And once we confirm this structure is the right one, we then push to purity tests. So with NMR, in addition to molecule ID, at the same time, it can give you some information as to the purity as well. And aside from NMR, we have HPLC or GC, and sometimes we do a titration or ion chromatography. Now and again, we use elemental analysis, and this can further support the chemical composition and provide some indication of the salt forms, which is really useful as well. So you can see we've got a lot of analytical techniques and instrumentation at our disposal, and from time to time we outsource things like X-ray. So in terms of the skills we use to ensure quality control, well, TRC chemists have got a strong background in organic chemistry as well as the analytical techniques. So this really helps with the wide variety of molecules our chemists work with. And at the same time, the quality assurance team, they've got an analytical or a synthetic background, and in most cases, both. And again, this helps them to navigate through the analytical results and to ensure the identity and purity are truly what they're supposed to be. So during this type of analysis of the identity data, no signal or result should conflict with the assumed chemical structure of the molecule. And if we do have these contradictions, then we need to resolve these before we release the molecule. And I'll talk a little bit about this on the next slide. Thanks. Yeah, so yeah, this brings me to consistency checks. And I want to touch briefly on what these are and how LGC use these to perform a final validation before product release. So during production, there's often a possibility to confirm data gathered by one technique by cross-referencing with the information derived from another. So for example, this HPLC chromatogram on the left here, and you also have this proton NMR on the right, and they were both generated from analysis of a reference standard for chlorobenzoic acid, and this is a bezafibrate impurity, so it's a lipid-lowering drug. And as you can see on the left circle, we have traces of an impurity, in the HPLC at quite low percentages. And then corresponding this, cross-referencing with the NMR, we can see the same impurity in exactly these trace quantities in the right circle. So confirming that the HPLC method, so these conditions reflect the purity correctly. And in fact, in this case, it's a hydroxyl derivative. So in general, it can be said that if we've got the appropriate experience in the team, using this approach of comparing different orthogonal methods, it can provide a powerful technique to ensure the correct identity impurity. And in this case, this is micromole data I'm showing, but TRC also perform this type of orthogonal testing. Thanks. And next slide, please. So I'm delighted to be joined by Dr. Tony Rantanen, he's been with the company since 2015, joined as a synthetic chemist and now is a quality team leader. So maintaining and onboarding all the skills and techniques required to support structure idea associated with such a big catalog is really challenging. And I'm pleased that Tony's agreed to talk you through onboarding some of these unusual techniques. So I'll hand over to you, Tony. Thanks.
Yeah, thanks, Joe, and hello, everybody. Good morning, good afternoon, good evening, wherever you happen to be joining in from. As Joe already touched on, so we do have quite a wide catalog, and you can envision how a catalog of 150,000 molecules, and of which we currently have 55,000 in stock, you can imagine what kind of challenges it poses for the structure determination of these compounds, as these structures range from really, really simple building blocks and research chemicals all the way to complex macrocyclic API impurities. So as Joe mentioned, we have our standard menu of testing that we use for all of our compounds. But in addition, we have some atypical techniques that we also use to augment really these perhaps more basic techniques, and in order to really ensure the quality of our materials so that the identity is correct and the purity is accurately represented. So a couple of these techniques are listed here as an example. So on the 15 nitrogen NMR, I'll be touching on a little bit more in a minute as in the form of a case study. 119 tin NMR, we have some tin containing organotin compounds, and the NMR is very useful to determine that tin is first of all present, and second to determine the purity of the material as well, as it's quite sensitive as an NMR technique. TGA stands for thermogravimetric analysis. So essentially you weigh out accurately a couple of megs of your material that you wish to analyze. We put it in an oven, on a little pan, so to say, and we heat it up to 1000 degrees, and we measure the weight loss as a function of temperature. So we use this for determination of volatiles as well as the determination of residual inorganics. And then additionally, we have high and low temperature NMR that we sometimes use as well. So these are just a couple of examples of perhaps the more atypical structure determination techniques that we do use, but I want to touch a bit more on the 15 nitrogen NMR in the form of a case study, as I personally found this quite interesting and exciting as well as a learning experience. Next slide, please. So this is an actual real-life case study from our synthetic labs. Now, unfortunately, I cannot show you the exact structure of the molecules, due to confidentiality. But the basic core of the molecule is shown here on the slide. So you see we have this aminotriazole that the chemist is reacting with the sulfonyl chloride, and the target molecule is sulfonylation on the exocyclic amino group. Now, the synthetic chemist had a chat with myself, and we brainstormed together what the problem was. And the problem was really the identity of the compound that was synthesized. So in this particular case, there was no exact literature reference available for this target compound. So we had really no analytical data to compare to. Now, the reference that we found together was from the Chemical and Pharmaceutical Bulletin in 2004. Now, they have taken this aminotriazole carboxylic acid, and they have synthesized via acylation with just acetyl chloride, and identified and isolated each of the 4 possible carboxamide products that can arise from this reaction. And additionally, they make the statement that I have italicized in the bottom. The exocyclic amino group on the acyl amino acids offers significant resistance to acylation and makes the synthesis of carboxamides difficult. Next slide, please. So now this, of course, poses a challenge to us, so to the chemists as well, as they were not sure if they had actually synthesized the right compound, and also for the quality team here at TRC for the material release. So we, of course, cannot release the material unless we are sure that the identity is correct. So these are the 4 possible structures that are identified here on the slide, and our task was to find out that which one of these structures is correct. Next slide, please. Now, again, as I'm not able to share the complete structure, but suffice to say that we didn't get any help from a standard set of 2D NMRs, so proton-proton COSY, proton-carbon HMBC, HSQC, as well as NOE experiments, as there are really no close by protons to compare them to have correlations with. Now, X-ray is an option, and of course, don't get me wrong, we do X-ray analysis as well. However, there is always a customer impact on this option, as we need to have the synthetic chemist grow crystals that would be suitable for diffraction analysis, as well as then, of course, we don't have an X-ray instrument here in-house, so we have to outsource the analysis. So this all translates into an extended lead time, which of course may have a negative impact to the end user. So it is an option that we do take. However, we do try to, of course, evaluate the structure in-house first before taking this step. Now, for this particular compound, as we did delve into the literature a bit deeper, we found in the same Chemical Pharmaceutical Bulletin paper the snapshot shown here below. And I would direct your attention to the point C, that it was a table of NMR chemical shifts, and the reference is to measure using proton to 15 nitrogen NMR GHMBC or GHSQC correlation experiments. Now, as mentioned above there on the slide, I do know proton-carbon HMBC, proton-carbon HSQC. However, I had not heard before of a proton-nitrogen HMBC or HSQC correlation experiments. Now, luckily, we had a very talented NMR specialist in-house, our NMR operator, Jamie. So I did go and have a chat with him about what could we do with this 15N NMR. And the thought in my mind was that, can we use 15 nitrogen NMR to somehow distinguish either from the chemical shifts, or using it somehow to identify the different chemical environments that each nitrogen atom on this molecule is facing. Next slide, please. Now, Jamie did confirm that the standard 15 nitrogen NMR, just like we would do 13C or 1H NMR, that would not work because, first of all, 15N, the stable isotope, the abundance is quite low, 0.4% approximately in nature. And also the sensitivity compared to a proton is only about 0.001. Now, in comparison, for example, carbon-13 NMR, the abundance is about 1.1%, and the sensitivity, if I remember correctly, is 0.02. Now, there are other more technical NMR aspects in play, but what this all translates to is that is the quote in the middle that Jamie told me, that in order to obtain a reasonable 15N NMR from, for example, cyclosporine, it would take more than 10 years of scanning. So this is obviously not really an option that we want to take. But Jamie did excitingly confirm that the nitrogen-proton correlation techniques that were referenced in the paper are possible. So 15N-HMBC, which studies the 2-to-3 bond nitrogen-proton correlation, or the direct nitrogen-to-proton correlation, respectively, in the form of an HSQC. And there on the right-hand side of the slide, you see just a table I pulled from the web that depicts the chemical shifts normally associated with 15N NMR. Next slide, please. Now, before using the material that the chemist had spent a lot of hours in synthesizing, we did do a test case. And you can see here a molecule, a reasonably simple oxidiazole that we had in stock that we had synthesized previously. So we did run the 15N HMBC correlation spectrum of it, and you can see it here on the slide on the left-hand side. Now I have labeled the 3 different nitrogens, and you can see the different correlations on the NMR spectrum as well. And if you remember that this HMBC technique is studying the nitrogen to proton correlations 2-to-3 bonds away. So the nitrogen labeled as A, it is expected to have 2 correlations, one with the directly bonded methyl group and the other one with the directly adjacent CH2 group. And those we see in the form of those 2 black dots on the NMR spectrum. The same thing with the nitrogen B. It is expected to have 2 correlations with both CH2 moieties in the molecule, and indeed we do see this also in the form of the 2 black dots on the spectrum. And then finally, the nitrogen C is expected to only have 1 correlation, again with the closest CH2 moiety. So to me, this was pretty exciting. Now, some of you may already know of this technique. It's not like that the technique is brand new and we have invented it here at TRC. The literature reference was from 2004, and I'm sure that this technique is used all over the world in different ways, but this was for me personally the first time that I had heard about it, and also the first time that I had actually seen it in action. So it's really nice to be able to correlate it with a real life example and see the NMR spectrum in front of you. So a really exciting example, and I was able to share this technique and the details with our analytical, with our production, and with our quality assurance teams. So now this is an added technique that we have added to our repertoire, and something we can use in the future to analyze those really difficult molecules. Next slide, please. Now, again, unfortunately, due to confidentiality reasons, I cannot share you with the actual spectrum, so you just have to take my word for it. But in a nutshell, the 15N HMBC and 15N HSQC, together with comparison of the chemical shifts, we were able to be a process of elimination. We were able to show that, yes, we had indeed synthesized the correct material, and we were able to release the compound for customer orders in a timely fashion, rather than having to wait for growing crystals for the X-ray diffraction analysis. Now, if there's any take-home messages from these couple of slides that I have presented, I would highlight two. One is the old adage that an hour spent in the library will save you a week in the lab, and that is absolutely true. Please do take the time to look into the literature and read them carefully, as you never know what kind of nuggets of information you can find from there. And the second, perhaps more importantly, is that at TRC, we don't shy away from investigating new techniques and perhaps thinking outside of the box when it comes to ensure quality. So we do pride ourselves as a learning organization. So, and this, I believe, is a really nice example to showcase that we did take time away from our day-to-day activities. Jamie did set the parameters for the NMR analysis, and we worked together in order to learn something new. And that's really always something that motivates me the most in my role, which is to learn some new and exciting things. But on that note, I'll pass the torch back to Joe and let him continue on with the presentation.
Thanks very much, Tony. That was really interesting. Thanks for going through that. Okay, next slide, please. Okay, so you know, on a theme of the literature, you know, further to Tony's comment, so you know, the literature can be useful, but you know, when you find problems in the literature, this can also be challenging, both from a kind of consumer perspective in knowing what to order in terms of impurities, and in addition, also in terms of making sure that we can manufacture the right impurity with confidence. So it's clear that finding a reliable synthetical route can be extremely time-saving. So in this particular case, a customer requested a targeted oncology drug impurity. So this is a dimer of Brunton's kinase inhibitor, ibrutinib. So the structure is shown in the top here. And this was listed in a patent referred to us by the customer as RS stereochemistry. So the TRC chemistry planning group investigated the synthetic data just as part of their normal planning procedures. And on reviewing the patent describing the compound, they discovered that the listed method to make the dimer used 2 fragments of a parent compound, and the stereocenters were not affected. And therefore, the recorded structure for this impurity must be wrong. And this, you know, potentially due to a clerical error in the patent or something like that, and scarily sort of propagated through to SciFinder database, leading to, you know, I guess like a request for the wrong molecule. But the chemistry planning group, they submitted a revised structure to the customer, shown here on the bottom, and this was accepted and ordered by them. And the new compound was manufactured and the correct structure confirmed by chiral methods. And had this not been spotted, then the customer would have had the wrong impurity, potentially leading to incorrect conclusions and possible safety or regulatory issues. Next slide, please. So our final case study, so this shows how shortfalls in quality of a reference standard can delay the release of a product to market at cost of time and money and resources. So we had a case where the customer detected 2 unknown impurity peaks in a shampoo, and you can see in the top in the middle there. So the shampoo was ready to sell on the market, but he couldn't release it because he didn't know what these 2 unknown peaks were. And from his in-house testing, he suspected a specific impurity to be the cause for these 2 peaks. And so he bought a standard and he performed some tests with extracted material from the shampoo, and nothing was really fitting. The retention time of the impurity was different to the standard, and the UV was a little bit different as well. And so from this work, he excluded the impurity as being the cause of this problem, and he came to LGC Micromol, TRC sister company, and he asked for custom services to solve the problem. So in these cases, the usual plan is for the team to perform isolations from the final dosage forms. So it could be tablets, ointments, or whatever, in this case a shampoo. And once we have the material, then the next step is to determine the structure by mass spec and two-dimensional NMR experiments. And the final goal within the group is synthesis of a reference material, which we can provide with a certain assay, and then the customer can solve his issues. So we went through this process in full, in this case, spiking more API into the sample to compensate for the low concentration. We then heat stressed and extracted and analyzed the impurity. And in fact, the results of all this analysis, so the investigations did confirm that the customer was right in his original analysis. So one of these impurities shown to be present in the product was correctly identified by the customer's in-house team, but he'd ruled it out. So this impurity was fortunately present in the TRC catalogue, and in this case, since it was required for an identity test, a qualitative standard was perfect from TRC, so this was used. And our team did actually compare this standard with the one the customer used on the bottom right here. So in this NMR data, you can see that the TRC standard on the bottom is very different from the impurity standard the customer purchased. So they wasted a lot of time and money and resources. In fact, they asked us if we'd could tell the difference here, but you know, I think the team had better things to do than analyze this sample. We suggested that he just throw it away. So yeah, this was just highlights the problems with using some of these impurities. Okay, so I hope this gives you you know, a good overview of I guess like the problems with you know, some standards, the importance of quality, and TRC's approach to purity and identity, the importance of this, and leading on to Tony's interesting case studies on the difficult structure ID techniques. We're of course happy to address any questions that you might have. Thank you.
Okay, well, thanks very much, Joe. Before we go to the questions, I think we've got a couple of poll questions just to get people going. So we're going to run 2 polls back to back, and I'll get Chris to launch the first one now. Okay, so the first product is, do you already use TRC products? And if you do, what are the major applications that you perform? Either peak identification, system suitability tests, verification of specific parameters, or something else, or you don't use TRC products at all. So I'll give you just a minute or so to cast your votes. And while we do that, I'll just remind you that you can submit questions at any time during the webinar. We've got a few coming in, but it'd be great to have more. This is the ideal time to get your questions about this topic answered by the experts that we have here. So do please get those questions into the question box at the bottom of the GoToWebinar panel. And if you missed at the start, you will get access to a recording of the webinar by email in the next few days. And a certificate of attendance will come in that same email. Just having a quick look at the poll. And we could do I'll leave it open for just a little bit longer. So again, thanks very much to the team from TRC and LGC for putting this webinar together. It's part of the problem solvers collection that we have at Chemistry World at the moment. So if you want to see all the other articles in that collection around solving some of these problems, then do head over to the Chemistry World website. Okay, I think we are ready to close that poll now. And we've got this. Do you want to see the results now? Yeah, so we've got a bit of a mix there. Quite a few people not using TRC products at all. So I guess that's a bit of an opportunity for you guys, Joe and Tony and the team. So hopefully we can convert some of those numbers to using the other products as a result of the webinar. Okay, so Chris, could you launch the second poll, please? Okay, so have you had problems with substandard impurities before, and what kind of problems have those caused for your business? Have you experienced any delays in product release, any increased production costs, problems in project planning, or invalidation of research findings? You can select any or all of those options if you want to. So please do get your votes going now. And as I said, don't forget to put your questions into the questions box while we wait. So as I was saying, this webinar is part of our Problem Solvers collection. That's been put together in collaboration with Notch Communications, and there's various articles on the Chemistry World website. If you go over to chemistryworld.com after the webinar, please don't go there now. But if you go over to the chemistryworld.com and look in the top bar, there's a bar that says collections. If you click there, that's where you'll find all of this information. Okay, so I think we've got some representative results there now. So let's close that second poll and we'll have a quick look at the results as well. So again, I think we've got quite a good mix of things, and clearly some people are experiencing various problems. So this is obviously an important topic for you guys. Joe, do you want to address any of those? Do you want to have a quick are those the kind of numbers that you might expect from those results?
Yeah, I guess quality is important to many different aspects of research and production. So it's not surprising to see quite a wide spectrum of problems.
Yeah. Okay, Chris, I think we can close the results now. Okay, so we'll go into the main kind of question and answer session now. We've got quite a few questions coming in. So I think the first one to ask, and I'll ask to Joe, is you mentioned there's a difference between qualitative and quantitative standards. Is there any difference between those in terms of how you prepare them, in terms of the challenges in getting them right? Can you give us a little bit more detail on that, please?
Yeah, sure. So there are regulatory considerations. So with a quantitative standard, quantitative standards produced by our sister company Micromol. So these are made in an ISO 17025 accredited lab, and these can be used to demonstrate compliance to various regulatory framework, such as ICH, Q3A, 3B, and the new 14, and also GMP, GLP. And from an analytical point of view, these sorts of quantitative standards are used for exact quantitation and validation of analytical methods. And Micromol will calculate residual water, volatiles, and inorganics, for example. So you have the true assay value. Okay.
Great. Okay. Well, thanks very much. And I guess to Tony, I mean, you mentioned X-ray as a kind of that. I guess coming. So I'm an organic PhD myself. So I see X-ray as a kind of as a gold standard almost of. You can definitely see that the thing is the right thing. How often do you need to go to that sort of level of to identify the standards?
Yep, the X-ray is definitely the ultimate way to prove that you do have the correct structure. We do it, I would say, a handful of times a year. It's really for those cases when the literature information may be contradicting, that there may be multiple reports reporting different things. When we simply using our standard techniques, or even the non-standard techniques, when we are simply not sure what was it that we made. So in those cases, we do rely on X-ray crystallography, and we get the results, and then we do know which structure we had. We actually did that just a couple of weeks ago. We had made 2 diastereomers on a steroid. And on discussing this with the synthetic chemist, we really did not know which one was which, and neither one was represented in the literature. So we did decide together that let's grow crystals, let's send them for X-ray, and we were able to unequivocally identify both of the diastereomers.
Brilliant. Okay. We've had a question from Sudhir who asks, and so on this, I think this is to Tony. So on the slides, you were talking about some sulfonamides derivatives, and he's sort of, so I'm guessing from experience, he's saying that those are very moisture sensitive. Did you have to be careful of, did you see any N-oxide impurity during your synthesis, and did you have to be careful, generally, do you have to be, particularly, do you have to look at, when you're looking at your synthesis, how do you know when you're going to have to, I mean, you're talking about impurities, you're kind of knowledgeable about impurities. Do you have an idea of predicting when the synthesis that you're using to make these compounds is likely to produce certain kinds of impurities?
Yeah, so we do have a couple of things. First of all, to find out the moisture sensitivity. Excuse me. So we mostly we do rely on the observations from our synthetic chemists. So they are the ones who are the experts. They are the ones who synthesize the compound, and their observations are really invaluable to determine the stability. And a lot of the information does come from them. Now, additionally, also there's of course certain classes of compounds that are known to be moisture sensitive, sulfonyl chlorides, acid chlorides, as the person asking the question identified sulfonamides. So definitely based on our own collective experience, we do have that in-house as well. And then finally, when we do perform our standard menu of testing, we do, as one of our standard proton NMR tests, we also perform a D2O wash. It may not always be needed, but we have found out that it quite often provides a lot of useful information. So that will, of course, give us a good indication of when we add a drop of deuterated water. If the NMR becomes a complete mess, it's a good indication that, yes, the compound might very well be moisture sensitive as well. So to this particular question on the sulfonamide, so yes, we did discuss with the synthetic chemist on the potential stability issues as well. However, this particular compound seemed to be quite stable. So neither the synthetic chemist nor the analytical chemist did observe any kind of instability towards moisture. And also on the mass spec as well, we did not observe this particular N-oxide either.
Okay. Great. Well, thanks very much. I guess the next one I think is for Joe. You mentioned very briefly that some of the micromole standards can be used for kind of regulatory substitutions. We've had a question from Stephen asking about GLP compliant standards to do GLP compliant studies. Can you give us a bit more information on that aspect of your catalog, please?
Yeah, the micromole products are many of them are validated for that, and yeah, I'm happy to kind of take the details of this offline and follow up and provide some more information, but I'm sure we can help with that.
Brilliant. Okay, and that is a general point. Thanks for all of the questions coming in, but yeah, if there are questions that we don't have time to get through to, then Joe and Tony have both said that they will. They may be able to follow up by email afterwards. I have one very, very technical question, which just struck me when I was looking at that NMR, Tony. So that you were doing a correlation experiment between proton and nitrogen NMR, and yep, you were looking at the correlations between the CHs and the nitrogen. I don't know if it was just me, but I couldn't. Why don't you see a signal for the directly bonded NH, or is that just not? Does that just not show up on the experiment that you did?
So, to my understanding, it would not show up on that particular experiment. However, there is the 15N1H HSQC, which would be specific for this particular task, what you mentioned. So that would show the protons directly bonded to the nitrogen. So that would be another. We did perform this experiment as it happens, and we did observe this correlation as well during this experiment. I just thought that the HMBC is maybe more exciting and informative.
Yeah, well, it definitely gives you a bit more information about the structure around the nitrogen, which I guess is what you're looking for. And I had one other question, which was so in Chemistry World, we had a story a little while ago about a compound called bosutinib, which there was a little bit of an issue around some standards. There was, as I understand it, there was a widely distributed and published structure, but the material being supplied by some suppliers had the wrong substitution pattern on one of the aromatic rings. How do you kind of go about making sure that that sort of thing doesn't happen with your compounds, or how do you go about identifying those kind of problems?
It's a good question, but also a very difficult question as well.
Absolutely.
So what we do is we of course we run our standard tests, and we have, as mentioned, a separate group who analyzes these test results. So really without any bias or without any stake in whether the compound will be released or whether the compound will be rejected. Now we do compare when possible, we do compare the analytical data to information available in the literature. Now, if the literature is wrong, those are the really difficult cases that may sometimes be impossible to fully identify. But what we do try to do is we try to look at all the data holistically, so to say. So we don't just rely on proton NMR, for example. We have the proton NMR, we have mass spec, we have CHN. We do the whole gamut of testing in order to ensure that the identity is correct. And I'm happy to say that most 99.9, and maybe add a couple of nines, of the compounds that we do release, that they are of the correct structure. Now, we have had absolutely a couple of cases where we have found out after the fact that we have synthesized the incorrect structure. Now, in those cases, we are of course beholden to our customers, and we do send notifications to our customers that, hey, we have made a mistake. We recall the material, and that is just our commitment to quality and our responsibility to our customers as well. Hope that answered your question.
Yeah, no, absolutely it does. Absolutely it does. I mean, some of these things very, very tricky, and especially if you can't, we talked about X-ray earlier, but some compounds don't crystallize. Some that you can't do X-ray and everything. So
and we can't do an X-ray from every single compound, of course. But this is why also the interaction between the different departments, analytical, synthetic chemistry, and then the quality assurance team, the interaction and the teamwork is very important. So we work together, we discuss, and then we come to a conclusion based on the discussions.
Yeah, brilliant. Okay, so I think we've probably got time for maybe one last question, which I guess is a bit of a general one. So maybe I'll get both of you to answer this question individually. We'll start with Joe. Can you tell us what's the biggest challenge of developing these new reference standards from your point of view?
Yeah, so I suppose it's I guess like coordinating all the teams, to ensure I guess like communication to ensure release of the correct compound.
Okay, sort of making sure that all of the things that we've just been talking about actually happen, and everyone's talking to each other to make sure you've got the least likelihood of any of those problems coming on. And Tony, from your point of view, what's the biggest challenge that you face from your side?
I would say that it's perhaps seeking the proper balance between the customer expectations around the lead time or delivery, as well as the quality. So quality is important, but equally well, very important, most important is also our customers and their expectations, and that they get the compound that they have ordered in their hands. So there are times when I have to balance the question, do I need to do more testing, or am I confident enough with what I have in hand to release the compound? And this is sometimes difficult to balance, and sometimes I have to make the decision that no, I'm not confident enough, and I need to request more testing just to be sure that the identity of the compound is correct.
Yeah, so I guess comes down back to that old kind of three-way challenge, as it can be good, fast, or cheap, and you can probably only have two of those.
That's right.
Absolutely. Okay, great. I think, guys, you've done a fantastic job answering the questions, so thank you very much once again. It's been really informative, and I hope that the audience has got answers to everything they want to do. Thanks again to LGC and TRC for sponsoring, and I'll mention again, this is part of our Problem Solvers collection that we've put together in collaboration with Notch Communications. Thanks to you, the audience, for attending and for your questions. A reminder, you will get an email with a link to the recording and a certificate of attendance in the next few days. If you liked this webinar and you'd like to see more, then do remember to check out chemistryworld.com/webinars to see what topics we've got coming up. And I hope to see you in another webinar very soon.